Imagine having a really terrible high-stress day at work. Your adrenaline is just pumping, you haven't slept well at all, and you happen to go in for a routine physical.
Right, which is always a fun time to get blood drawn.
Exactly. So a few hours later, you look at your lab results, and your white blood cell count is double the normal upper limit. And the chart has this bold red flag next to the word leukocytosis.
Oh, yeah. That's when the panic sets in.
You immediately assume you have some massive systemic infection, right? But here's the biological trick. Your body hasn't actually produced a single new white blood cell.
Not one.
That spiked count is an illusion. It's basically a temporary shell game played by your own circulatory system.
It really is one of the most common optical illusions in modern medicine. We look at a complete blood count of CBC, and we just assume we're seeing this fixed, immutable snapshot of our biology.
Like it's an absolute truth.
Right, exactly. We treat the numbers as absolute truth.
Yeah.
But a blood draw is really just capturing a river at a single, isolated moment in time.
And today, we are going to decode the secret language printed on that medical chart sitting in your filing cabinet.
The one you probably Google at 2 a.m.?
Yeah, the one that keeps you up at night. We're looking at the suffixes as penia and cytosis. We are taking a whole stack of medical history, hematology research, and cellular biology sources to really understand the delicate balancing act happening inside your vessels right this second.
It's all about the extremes, really. Too few cells and too many.
Okay, let's unpack this. Because honestly, turning this wall of unpronounceable words into a mechanical understanding of how your body regulates its extremes, well, it completely changes the way you look at your own health.
It really does. And to understand those extremes, we have to define the opposing ends of this biological seesaw, which takes us all the way back to classical Greek.
Of course it does. It always goes back to Greek.
Always. So these are ancient modular word-building elements. The suffix penia, it comes from the Greek word pitenia, which translates to poverty or lack.
So when you attach that to a specific root word, it denotes a deficiency.
Exactly.
So neutropenia is a poverty of neutrophils. Lymphopenia is a lack of lymphocytes. Thrombocytopenia is a lack of platelets, which are clinically called thrombocytes.
You've got it. That covers the too few side of the equation.
Right.
Now, on the other end, we have air cytosis. This combines chitos, meaning hollow vessel or cell, with erdososis, which is a Greek suffix meaning a condition of or an abnormal process.
Okay, so it's a condition of cells.
Right. And in the clinical realm, a cytosis usually acts as the mirror image of leukopenia. It signals an abnormal increase in the number of a specific cell type circulating in your bloodstream.
Like leukocytosis.
Yeah, meaning an elevated overall white blood cell count. Or thrombocytosis, which indicates an elevated platelet count.
Okay, but I want to push back on that clean dichotomy for a second.
Oh, go for it.
Because I've read papers discussing phagocytosis, which is the process of a cell engulfing or eating debris, right?
Right.
Or penocytosis, which is a cell taking in fluids, like cell drinking. Neither of those terms refers to a high count of eating or drinking cells. It kind of seems like the suffix is doing double duty.
Ah, yes. You identified the exact semantic curveball that causes so much confusion across different medical disciplines.
It's super confusing.
It is, because the definition of cytosis actually splits depending on the context of the researcher.
Oh, really?
Yeah. So a hematologist, a blood specialist, hears cytosis and immediately thinks in terms of a quantitative count, too many cells.
Right.
But a cell biologist hears that exact same suffix and thinks of a mechanism.
Like endocytosis.
Exactly. Endocytosis, bringing material into the cell, or exocytosis, expelling it. Those use the older, broader definition of a process involving cells. It tells you absolutely nothing about the total cellular population in the body.
Okay, that makes sense. And the leukopenia side of the seesaw isn't strictly confined to cell counts either, right? If we look at a term like osteopenia.
Right.
That describes a reduced bone mineral density. And bone tissue contains very few living cells relative to its calcified matrix.
Or cytopenia, which describes an iron deficiency.
There are no cells involved there at all.
None. Just a mineral shortage. The truest meaning of these suffixes is simply deficiency and abnormal condition. It just so happens that in the modern era, cell counts are the most frequent place we encounter them.
Which introduces this massive historical bottleneck in the sources.
Oh, the timeline here is fascinating.
Yeah. Because having the vocabulary for a poverty of cells in the early 19th century, it's basically like having the word for a traffic jam before the combustion engine was invented.
That is a perfect way to put it.
The diagnostic language existed, but the ability to measure the disease didn't. I mean, you can't declare a specific cell line is deficient if you physically cannot tell the cells apart.
Right, because before the 1870s, physicians would just look at a blood smear under a crude microscope and essentially shrug and say, this blood looks
It's
pathological.
just sick blood.
Pretty much. They could see red cells and maybe a few translucent white cells. But the white cells were just this homogeneous, undifferentiated blob.
So there's no way to count a specific subtype.
Zero. The breakthrough that solved this came from a German physician named Paul Ehrlich in his 1878 doctoral thesis.
So what did he do? Did he just get better microscopes?
No, it was actually about chemistry. Ehrlich was fascinated by the nascent synthetic dye industry. He began applying various aniline dyes to blood smears, statistically testing acidic versus basic chemical dyes.
Like cloaking dyes.
Essentially, yeah. And what he discovered revolutionized immunology. He found that the internal structures of white blood cells, their microscopic granules, had completely different chemical affinities.
So like some granules loved the acidic dye and some loved the basic dye.
Exactly. And some remained neutral. So suddenly this homogeneous translucent blob split into distinct characters, pink and blue dots everywhere.
Wow. And those visual differences gave us the foundational names we still use today, right?
Yes, they did. Eosinophils are named because their granules readily absorb eosin, an acidic red dye.
Okay.
Basophils absorb basic blue dyes. Neutrophils absorb both to create a neutral color.
Hence, neutrophil.
Right. So Ehrlich gave us the differential count. A physician could finally quantify exactly how many of each specific cell type were present in the blood.
But what's incredible to me is that while Ehrlich provided the visual map, the modern clinical weight of penia and cytosis was actually cemented by engineering in the mid-20th century.
What's fascinating here is how automation changed everything. I'm talking about the Coulter counter.
Right, in the 1950s.
Yeah, Wallace Coulter patented a technology based on electrical impedance. So instead of a technician hunched over a microscope for hours, manually counting pink and blue dots on a glass slide.
Which sounds exhausting.
Oh, highly prone to error, too. But instead of that, a machine drew blood through a microscopic pore. And because cells are poor conductors of electricity, every time a cell passed through the pore, it briefly interrupted an electrical current.
So the machine could just count the interruptions.
Exactly. It counted the interruptions, sized the cells based on the magnitude of the electrical spike, and categorized them instantly. It transformed the CBC from a laborious manual task into a cheap, automated global standard.
And once medicine had the technology to routinely count these cells, the clinical stakes of a quantitative shift became glaringly apparent.
Oh, completely. Your body maintains each cell line within a strict reference range, because dropping below that threshold removes a critical piece of your physiological infrastructure.
So let's look at the mechanical stakes of the leukopenia side, specifically neutropenia in the context of oncology.
Right, a very common scenario.
Because neutrophils are the innate immune system's primary responders to bacterial pathogens.
Right, your first line of defense.
And a healthy absolute neutrophil count, or ANC, usually sits above 1,500 cells per microliter. But standard cytotoxic chemotherapy routinely crashes that number.
Yeah, and the reason for that is chemotherapy is largely designed to disrupt the cell cycle in rapidly dividing tissue. The whole goal is to halt tumor growth.
Right.
However, the myeloid progenitor cells in your bone marrow, the stem cells that constantly divide to supply your daily billions of new neutrophils, they are just as vulnerable to the drug as the cancer cells.
So the bone marrow just gets suppressed as collateral damage.
Exactly. If the ANC drops below 500, you hit severe neutropenia. And without those first responders, a routine pathogen, something your body would normally clear without a second thought, can replicate unchecked.
Which leads to febrile neutropenia.
Right, which is an absolute medical emergency. It triggers immediate medical interventions like administering IV antibiotics and GCSF, granulocyte colony stimulating factor. It is a synthesized version of a naturally occurring glycoprotein.
And what does that do exactly?
When injected, it binds to specific receptors on those dormant or suppressed stem cells in the marrow. It essentially sends an aggressive chemical signal overriding the suppression, forcing the marrow to rapidly proliferate and mature neutrophils to repopulate the blood.
Wow. But that's an acquired deficiency from a drug. The sources also highlight Kostman syndrome, where the aphipene is actually hardwired into the genetics from birth.
Yeah, Rolf Kostman, a Swedish pediatrician, documented this back in 1956. We now know severe congenital neutropenia is often caused by mutations in specific genes, like the ELANE gene, which codes for neutrophil elastase.
So what happens when that gene is mutated?
The mutation causes misfolded proteins to accumulate inside the developing neutrophil. The cellular machinery detects this structural failure and triggers apoptosis.
Programmed cell death.
Right? The neutrophils effectively commit suicide in the bone marrow before they can ever mature and enter the bloodstream.
That is wild. Okay, so flipping the biological seesaw, what are the mechanical stakes when the marrow overproduces? Let's look at polycythemia vera.
Polycythemia vera is a rare blood cancer where the bone marrow churns out an excessive number of red blood cells. It pushes the blood volume and viscosity to really dangerous levels.
The blood gets too thick.
Exactly, it becomes thick, drastically increasing the risk of thrombotic events like strokes or heart attacks. And for decades, it was just a descriptive diagnosis. Too many red cells.
But in 2005, researchers found the mechanical switch causing the problem, right? The JAK2 V6MTF mutation.
Yes, JAK2 is a kinase, which is a type of enzyme that acts as a signaling switch. Normally, for your marrow to produce red blood cells, a hormone called erythropoietin must bind to a receptor on the outside of the cell.
Okay, so it needs a key.
Right. This binding activates the JAK2 kinase inside the cell, sending the signal to divide. But the V617F mutation physically alters the shape of the JAK2 protein, essentially removing its autoinhibition domain.
So it's basically like hot wiring a car. You don't even need the key, the hormone to turn the engine on.
That's a perfect analogy. The kinase is stuck in the active position, constantly telling the marrow to proliferate, regardless of what the body actually needs.
That molecular discovery completely transformed a vague cytosis condition into a targeted genetic disease.
It did.
But we should probably address the terminology mess you might see on a chart alongside these conditions, because the suffixes aren't always uniformly applied.
Oh, not at all. It's a bit of a historical mess.
Yeah, like the red cell excess is polycythemia, not erythrocytosis. And we use anemia instead of erythropenia. How do we even map these neighboring terms?
It really comes down to historical usage versus strict nomenclature. Anemia, for example, literally means without blood and RA plus anemia. The suffix atemia refers broadly to a blood condition.
Okay.
And when it comes to the white blood cell subsets we mentioned earlier, the granulocytes, we use the suffix philia, meaning attraction or affinity.
So a high eosinophil count is eosinophilia.
Yeah, not eosinocytosis, even though the biological concept is literally identical to lymphocytosis.
And what about cythemia?
That is generally reserved to indicate a primary marrow-driven overproduction, like essential thrombocythemia, differentiating it from just a secondary reaction to a random infection.
Okay, that makes sense.
And then there is pancytopenia, where the prefix pan means all. This is the simultaneous crash of red cells, white cells, and platelets. It points mechanically to a complete bone marrow failure rather than an isolated cell line issue.
Okay, which brings us all the way back to the scenario we discussed at the very beginning. The idea that reading a CBC is just simple math. Too high, too low, identify the problem.
Right.
But as we said, the numbers can be deceiving. Let's dig into the mechanical reality of stress leukocytosis, where your white count spikes without any new cell production. How does that illusion actually work?
It comes down to fluid dynamics and cellular adhesion. The neutrophils in your body are divided into two distinct pools. There is the circulating pool, which flows freely in the center of the blood vessel.
And that's the blood we draw with a needle.
Exactly. But there is also a marginated pool. These neutrophils are physically tethered to the endothelial cells lining the blood vessel walls using these adhesion molecules called selectins and integrins.
So they're essentially parked along the shoulder of the highway, just waiting to be deployed to local tissue.
Perfect way to visualize it. Now, when you experience acute stress trauma, extreme exercise, or severe emotional distress, your body releases epinephrine.
Adrenaline.
Right. The epinephrine does two things. First, it increases cardiac output, changing the physical shear stress of the blood flowing through the vessels. Second, it downregulates those adhesion molecules.
Oh, wow. So they let go.
The selectins release their grip, and this massive reserve of marginated neutrophils is suddenly swept off the vessel walls and into the main circulation.
So the total number of troops in the body didn't increase. They just moved from the barracks to the active battlefield where the needle could actually find them.
Exactly. The count doubles, but it's a temporary mechanical shift. It's not a pathological infection.
Which highlights a fundamental rule of hematology from the sources. A cytopenia or a cytosis is a finding. It is not a diagnosis.
Right. Take ITP, or immune thrombocytopenic purpura. The CBC will show thrombocytopenia dangerously low platelets, but the suffix doesn't explain the mechanism.
The low count is just the aftermath of the actual disease process.
Exactly.
Hmm.
In ITP, your immune system basically malfunctions and produces autoantibodies. These antibodies mistakenly target normal proteins on the surface of your own platelets.
And then what happens?
Well, as these antibody-coated platelets circulate through the spleen, splenic macrophages recognize the antibodies and destroy the platelets. The bone marrow is actually producing plenty of platelets, but they're being actively consumed in the periphery.
So the word thrombocytopenia points to the crime scene. It doesn't identify the culprit.
Exactly.
So if the numbers can trick us through physiological mechanisms like marginated pools, the sources show they can also trick us systematically.
This is where it gets really concerning.
Yeah. What happens when the baseline we use to define normal is structurally flawed from the start?
The most consequential example of this is a physiological state known as benign ethnic neutropenia, or BEN.
Right. Because for decades, the standard reference ranges defining a healthy neutrophil count were calibrated using blood samples predominantly from white European-descended populations.
If that was the baseline.
But global hematology eventually proved that many people of African descent, as well as certain Middle Eastern and Mediterranean populations, naturally maintain an absolute neutrophil count between 1,000 and 1,500.
And under the standard reference range, a count of 1,200 is automatically flagged with the pythopenia suffix. It is classified as an abnormal deficiency.
But for these individuals, it is their healthy baseline.
Right, carrying zero increased risk of opportunistic infections. The mechanism behind this lower circulating count is fascinating, though, because it links directly to the Duffy null genotype.
The Duffy antigen is a protein receptor located on the surface of red blood cells, right?
Correct. And it just so happens that Plasmodium vivax, which is a widespread species of malaria parasite, uses that specific Duffy receptor as its primary gateway to invade and infect the red blood cell.
So evolutionary pressure in malaria endemic regions selected for the Duffy null trait.
Exactly. Individuals with this trait simply do not express that receptor. They lack the gateway, granting them natural resistance to that specific form of malaria.
But wait, how does missing a receptor on a red blood cell lower the count of white blood cells in the circulation? Like mechanically?
It's a great question. The Duffy antigen doesn't just sit on red blood cells doing nothing. It also acts as a regulator for chemokines, which are signaling proteins in the blood.
Okay.
The current understanding is that lacking the Duffy antigen alters how these chemokines distribute neutrophils. The neutrophils aren't actually missing, they're just heavily sequestered in tissue reservoirs like the spleen, rather than floating freely in the bloodstream.
Ah, I see. But the clinical fallout of applying a rigid single population baseline to a diverse global population was severe.
It was devastating for many.
Yeah, healthy individuals had their blood flagged as abnormal, which led to them being systematically excluded from clinical trials.
Right.
And in oncology, healthy patients had life-saving chemotherapy doses reduced or even delayed because physicians saw the leukopenia flag and feared their immune system was failing.
When in reality, their marrow reserves were perfectly robust. It perfectly illustrates the danger of treating a statistical cutoff as absolute biological law. We actually see a similar debate in the sources regarding osteopenia.
Oh, yeah. The WHO established the definition of osteopenia in 1994, utilizing bone density DXA scans, and they defined it as a T-score between Medish 1 and Medical 2.5.
But the original intent of that definition was purely for epidemiological research. It was to study population trends over time.
Just a statistical category.
Exactly. It was meant to describe the middle of a bell curve, not a disease state requiring intervention.
Yet once the suffix was applied, it quickly morphed into a clinical target. The medicalization of a statistical borderline created this massive new population of patients.
Oh, overnight.
Millions of people who were not strictly osteoporotic were suddenly being prescribed bisphosphonate drugs to treat a state that, for many, was just a natural consequence of aging.
Because when you attach a suffix meaning deficiency to a physiological measurement, the medical system is primed to correct it. We have to be really rigorous in proving that the correction actually outpaces the risk of the treatment.
So, synthesizing all of this, we've moved from Ehrlich's chemical dyes in the 1800s to the electrical impedance of automated counters to the genetic misfolding of proteins. Why does an intricate understanding of this vocabulary matter for you today?
It matters because these suffixes are the active language of modern global health crises.
Like COVID-19.
Exactly. Look at the early days of the pandemic. Lymphopenia, a sharp, sudden drop in circulating lymphocytes, emerged as one of the most reliable prognostic markers for severe disease.
Because the virus was targeting them.
Yeah, the SARS-CoV-2 virus wasn't just infecting respiratory tissue. It was inducing widespread apoptosis in lymphocytes, basically crippling the adaptive immune system's ability to coordinate a defense. A 19th century descriptive suffix literally became the focal point of triage in modern ICUs.
And then in 2021, we saw the emergence of VITT, or vaccine-induced immune thrombocytopenia.
Right, which was an exceedingly rare but critical complication linked to certain adenoviral vector COVID vaccines. The mechanism was incredibly complex.
It was a clotting issue, right?
Sort of both. Patients developed rogue autoantibodies against a complex formed by platelet factor IV. These antibodies massively activated the platelets, causing catastrophic clotting throughout the body.
Oh, wow.
But because all the platelets were being actively consumed in these widespread clots, the CBC showed a profound thrombocytopenia.
So the blood test showed a lack of platelets, but the actual pathology was an aggressive, fatal overactivation of them.
Exactly. And tracking the mechanics of that specific cytopenia dictated global vaccine policy and regulatory approvals in real time. These are not just academic roots to memorize for a biology exam.
No, clearly not.
They are the scaffolding upon which we build our entire understanding of cellular failure.
So if you pull out your latest lab results and look at that column of words ending in cytopenia and cytosis, you are no longer just looking at a confusing math problem.
Not anymore.
You are looking at the quantitative legacy of aniline dyes and Coulter pores. You're looking at a dynamic, shifting ecosystem where stress, genetics, and your environment dictate the flow of millions of cells every single second.
And it leaves us with a really compelling question to analyze moving forward.
What's that?
Well, if rigid definitions of a deficiency, like the neutrophil baselines that ignore the Duffy null genotype, were accidentally biased by geography and genetics for decades, causing tangible clinical harm.
Right.
What other standard cutoffs in modern diagnostic medicine are built on flawed statistical bell curves? We may still be staring at the numbers, completely misunderstanding the mechanics of the river.